Hello everyone and welcome to this video where I'm gonna try and explain to you what is Western Blot. As you probably already know, Western Blot is a widely used laboratory method to detect specific proteins from among a mixture of proteins. This technique also allows us to evaluate the size of a protein of interest and to identify variations in the amount of protein expression. Basically, the Western Blot procedure can be divided in 4 steps that consist in sample preparation, protein separation, electrotransfer, and immunodetection. The very first step required to perform a Western Blot is to extract the proteins from tissues or cell cultures. We will follow a specific protocol depending on the protein of interest type, but it's crucial, whatever it will be the protocol, to keep the sample in ice all the time in order to avoid protein degradation. Then, we have to determine the protein concentration of each lysate to know how much protein we have to load in each gel well. We will add an equal volume of loading buffer, which contains SDS that is used to denature proteins and make them negatively charged. In this manner, each protein will migrate in the electrophoretic field in a measure proportional to its length. The loading buffer also contains beta-mercaptoethanol that is used to break disulfide bonds, glycerol that increase the density of the sample relative to the surrounding running buffer and make it easier to load the sample in the well, and bromophenol blue that is a dye used to follow the run of protein sample on the gel. This mixture is heated in agitation at 95 degrees during 4 minutes. The second step is the protein separation according to their size. We normally use SDS polyacrylamide gels that we prepare in the lab, and that typically consists on a mixture of acrylamide bisacrylamide, a denatural SDS, water, and a buffer with an adjust pH. Then, ammonium persulfate and timid are added to initiate polymerization. The polymerization reaction creates a gel because the bisacrylamide forms crosslinks between 2 acrylamide molecules, forming a pore. The acrylamide concentration can be modified according to the size of our protein of interest. In this way, lower percentage gels are better for resolving very high molecular weight proteins, while higher percentage of acrylamide are needed to resolve smaller proteins. There are also commercial already prepared gels that contain a gradient percentage of acrylamide, and that makes them very useful when we don't know the size of the protein we want to separate. Gels are usually polymerized between 2 glass plates in a gel caster and with a comb insert at the top to create the sample wells. Once the gel has polymerized, it has to be mounted in the electrophoresis chamber using binder clips. We pour running buffer into the upper and lower chambers and remove the comb from the gels. If any air bubbles or small pieces of the gel are present into the wells, they have to be eliminated. The following step is to load the previously prepared sample and the molecular weight marker in the wells. Then, we turn on the power supply, we fix a voltage, and run the gel until the dye in the sample buffer reaches the bottom of the gel. The third step is the transfer of the separated proteins from the gel onto a membrane. This is possible since the proteins have been bound with negative electrical charge provided by SDS. The 2 most common electrotransfer methods are wet and semi-dry transfer. Although the semi-dry one is faster, here we use wet transfer since it has less tendency to failure and it's recommended for large proteins. According to the membrane for Western Blot, there are also two basic membranes available, nitrocellulose and PVDF. PVDF has a higher capacity than nitrocellulose, but needs more careful pretreatment. We need to cut the membrane to the appropriate size, usually a little larger than the gel, to soak it in methanol for about 30 seconds, wash it in distilled water for about 1 minute, and finally incubate the membranes in transfer buffer for 5 minutes. To carry out the wet transfer, we have to build the so-called sandwich, formed by sponge, paper, gel, membrane, paper, and sponge again. This sandwich is clamped tightly with no air bubbles within it. It's important to keep all the components of this structure in transfer buffer all the time to prevent them from drying out. We also have to keep in mind that once in the chamber, the gel has to be the closest to the negative electrode and the membrane closest to the positive one. Then we fix the amperage and keep the transfer at 4 degrees for 2 hours. The fourth and final step is immunodetection. That is the use of specific antibodies to detect the protein of interest. The membrane with transferred proteins has to be blocked to prevent non-specific binding of antibodies to the membrane. There are several solutions that we can use to block the membrane. The most commonly ones are 1 to 5% of bovine serum albumin in TTBS or 1 to 10% skim milk in TTBS. Skim milk solution is more effective and that's why it's more frequently used. However, it contains the casein phosphatase and therefore is not suitable for the detection of phosphoproteins. Blocking time can vary, being at least 1 hour at room temperature, although the membrane could also be left blocking overnight at 4 degrees. After the blockade, we have to wash the membrane for 10 minutes with TTBS and repeat this step 3 times. Now is the time for primary antibody incubation. This antibody is specific to the target protein and will bind it on the membrane. In Western Blot, primary antibodies should be validated before use to select the optimal condition such as dilutions or incubation time. The primary antibodies can be monoclonal or polyclonal, and the choice of one or another depends on the antigen to be detected. Monoclonal antibodies recognize single specific antigenic epitope, and that's why they are more specific than polyclonal ones, which recognize more epitope. These labs often have higher affinity. After the appropriate incubation time, the unbound antibody has to be removed. so we wash the membrane 3 times with TTBS for 10 minutes each. Immediately after, the membrane is exposed to a specific enzyme-conjugated secondary antibody that binds the primary one. The most popular secondary antibodies are anti-mouse and anti-rabbit immune globulin, since the host species for primary antibodies are mainly mouse and rabbit. Finally, once the secondary antibody has been incubated, we'll rise the membrane 3 times again, and our proteins will be ready to be detected. As shown in the image, the secondary antibody forms a complex with an enzyme that, in the presence of a specific substrate, will create a detection signal. Alkaline phosphatase and horseradish peroxidase are the 2 most commonly used enzymes. The first one catalyzes a colorimetric reaction, while the horseradish peroxidase, that is the one that we normally use, catalyzes a chemiluminescent reaction. In the presence of the detection reagent that is formed by luminol and hydrogen peroxide, the chemical reaction produces light that can be recorded and that allows us to see the bands corresponding to our proteins.